Sterile loop or a cotton swab to pick colonies from an overnight culture on non-selective media. Use several morphologically similar colonies to avoid an atypical variant. Suspend the colonies in saline and mix to an even turbidity. The density of the inoculum suspension should be equal to 0.5 McFarland standard. It is recommended that a photometric device is used to measure the density of the suspension. Alternatively, the density can be compared visually to an 0.5 McFarland turbidity standard. If the density is too low, add more bacteria. If the density is too high, add more saline. If Streptococcus pneumoniae is suspended from a chocolate agar plate and not a blood agar plate, the inoculum must be equivalent to a McFarland 1 turbidity standard. Use the adjusted inoculum suspension optimally within 15 minutes of preparation and always within 60 minutes of preparation.
McFarland standard is a chemical solution of barium chloride and sulfuric acid. The reaction between these two chemicals results in the production of a fine precipitate, barium sulfate. When shaken well, the turbidity of a McFarland standard is visually comparable to a bacterial suspension of known concentration as indicated here. The accuracy of the density of McFarland standards can be checked using a spectrophotometer with a 1 cm light path. A 0.5 McFarland standard has an absorbance reading of 0.08 to 0.1 at the 625 nanometer. Alternatively, adjusting a bacterial suspension to the same turbidity and preparing serial 10-fold dilutions can verify the accuracy of a McFarland standard by performing a plate count of the dilution and ensuring that the suspension gives a representative colony count.